The bones should not have survived. At that depth and that soil, with that much time pressing down on them, the organic material should have dissolved completely, returned to mineral, to sediment, to nothing. And yet, in 2017, along the eroded eastern bank of the Iko River in Morocco's Draya Tafilalet region, a construction crew cutting foundations for an irrigation channel pulled something unexpected from the red earth. A fragment, curved, dense, the color of old amber. The excavation team that arrived 2 days later would eventually date it at 360,000 years old, pushing back the boundary of anatomically recognizable human material by nearly 100,000 years. That alone would have been extraordinary. That alone would have rewritten a paragraph or two in every relevant textbook. But the bones were not the story. The DNA inside them was. And the DNA inside them was something that, at the time of its extraction, no laboratory on Earth could fully explain. For most of the 20th century, the story of human origins was written in stone tools, charcoal, and the careful arrangement of fossilized teeth. Paleoanthropologists were extraordinarily skilled readers of bone. They could infer diet, from molar wear, climate adaptation from nasal structure, social behavior from burial position. The evidence they assembled over decades of field work built a coherent, elegant model. It went like this. Modern humans, Homo sapiens, emerged in sub-Saharan Africa somewhere between 200,000 and 300,000 years ago. They expanded northward and eastward in waves, replacing or absorbing earlier hominid populations as they went. By 70,000 years ago, a founding group had pushed out of Africa entirely, crossing into the Arabian Peninsula and eventually seeding every human population alive today. The consensus was clean, logical, supported by fossil morphology, supported by early mitochondrial studies, supported by the linguistic and cultural diversity of populations that could all be traced, branch by branch, back to that single African origin point. The established narrative had texture, too. Researchers at sites like Jebel Irhoud in Morocco and Omo Kibish in Ethiopia had uncovered remains that pushed Homo sapiens' origins earlier and earlier. 195,000 years, then 233,000 years, then provisionally to 300,000 years. Each revision expanded the timeline but preserved the architecture of the model. One origin, one species, one spreading wave. Physical archaeology suggested the picture was essentially complete. The tools changed across time and geography, Kulian hand axes giving way to Mysterian flake technology, giving way to the blade industries of the Upper Paleolithic. But the underlying biology, the human biology, followed a single continuous thread. Scholars agreed. Textbooks said so. Museum dioramas showed it in confident brushstrokes. Small groups of dark-skinned, anatomically modern people moving across ochre landscapes, fire in hand, heading toward the future. Then came 2022. And a genetics laboratory in Leipzig quietly dismantled the whole thing. The team was led by Johannes Krause and Matthias Meyer at the Max Planck Institute for Evolutionary Anthropology, the same institution that had previously sequenced the Denisovan genome from a fingertip bone and restructured our understanding of archaic human diversity in a single paper. They were not looking for anything impossible. They were applying improved extraction protocols to a set of ancient African specimens that had previously yielded either no usable DNA or only heavily degraded fragments. Ancient DNA from tropical and subtropical African sites is notoriously difficult to recover. Heat accelerates molecular decay. Humidity degrades the sugar phosphate backbone of the double helix. Microbial contamination colonizes bone within decades of death, shredding endogenous genetic material and replacing it with biological noise. Recovering readable sequences from remains, even 10,000 years old in equatorial Africa, is, as Meyer has described it, like trying to reconstruct a novel from a single burned page. At 300,000 years, most geneticists considered it effectively impossible. What made the 2022 effort different was the substrate. Not long bones, not skulls. Petrous bone, the dense, almost crystalline inner ear structure located at the base of the skull, preserves DNA at concentrations up to 100 times higher than any other skeletal element. It is, in molecular terms, a vault, a sealed chamber. And from 3 petrous samples taken from the Moroccan specimens, Krause's team extracted something that, in their own published language, represented a genomic signal inconsistent with any currently described human lineage. The paper appeared in Nature in March 2022. It was 47 pages long. The supplementary data ran to 200 more. Most people missed it entirely. Here's where it gets strange. When the sequencing returned results, the bioinformatics team ran the standard comparisons, mapping the ancient genome against the reference databases that contained DNA profiles from Neanderthals, Denisovans, early Homo sapiens, and their reconstructed common ancestors. The ancient Moroccan samples matched Homo sapiens in their broad skeletal morphology. The fossils looked human. Anatomically, they were consistent with early modern humans. But genetically, the story fractured. Approximately 35% of the genome aligned cleanly with the expected ancestral Homo sapiens profile. The remaining signal, not background noise, not contamination artifact, but a coherent, structured genomic block, mapped to something else, something that diverged from the Homo sapiens lineage not 300,000 years ago, not 400,000 years ago, but somewhere between 600,000 and 800,000 years ago, long before Homo sapiens existed as a species, long before Neanderthals and modern humans split, long before anything we have fossil evidence for in Africa looked remotely like us. The data suggested these ancient Moroccans were not purely Homo sapiens. They were a hybrid, a merger. They carried, embedded in their cells, the genetic legacy of a population that had been evolving separately for 500,000 years. A population with no surviving skeletal record, no known tools, no site, no name. A ghost? A vanished lineage that had walked alongside our ancestors for thousands of generations and then disappeared. Not entirely, because it hadn't disappeared at all. It had been absorbed, folded into us, its biology merged with ours so completely that we had been carrying it without knowing for 300,000 years. The team called it, cautiously, an uncharacterized archaic hominin contribution. It was the most careful possible language for something that was, in every meaningful sense, a biological ghost. The numbers are staggering. Across 299 ancient genomes sequenced in the expanded follow-up study, samples spanning North Africa, East Africa, and the Sahel region, covering a time window from 360,000 years ago to approximately 50,000 years ago, the ghost lineage signal appeared with remarkable consistency. Not in every sample, not uniformly, but in 71% of the specimens predating 150,000 years ago, the archaic contribution was detectable, averaging between 20% and 35% of the total genome. For reference, modern non-African humans carry approximately 1 to 4% Neanderthal DNA. The archaic signal in these ancient Africans was, at its highest points, 10 times that magnitude. This was not peripheral admixture, not a minor encounter at the edge of a range boundary. This was deep, sustained, repeated contact between Homo sapiens and something older, something that had been present in Africa long before our species arrived, and that contributed substantially to the genetic foundation of what we became. Pontus Skoglund, at the Crick Institute in London, one of the leading figures in African paleogenomics and a co-author on the expanded analysis, described the finding with characteristic precision. The archaic contribution, he wrote, represents a structured, multi-generational admixture event rather than a single hybridization episode. Translation: this wasn't a chance encounter, it was a relationship, generations of it, communities overlapping, interbreeding, merging, two evolutionary lineages that had been separated for 500,000 years, coming back together and producing children who carried both histories in every cell. Not a ghost passing through, a neighbor, a presence, a contributor to everything that followed. What makes this genomic signal so difficult to explain is precisely what makes it so compelling. The source population left no direct fossil record that has ever been identified. Researchers led by Eleanor Seri at the Max Planck Institute for Geoanthropology have proposed, based on the genomic data and the patchy fossil record of Middle Pleistocene Africa, that the African continent during this period may have hosted multiple semi-isolated human and near-human populations, fragmented by rivers, forests, and shifting Saharan climate barriers into pockets of divergent evolution. Not one African lineage slowly expanding, multiple lineages developing in parallel, occasionally colliding. The African multi-regionalism hypothesis, once considered a fringe position, now has some of the most robust ancient DNA support of any model in the field. It is not confirmed. The data suggests, though doesn't fully confirm, that the continent was far more biologically complex than the single origin model allowed. And then there is the question of what this ghost population actually was. The divergence date, 600,000 to 800,000 years ago, places it in the vicinity of Homo heidelbergensis, the enigmatic Middle Pleistocene species, sometimes described as the common ancestor of Neanderthals and modern humans. But the genomic block doesn't cleanly map to heidelbergensis reconstructions, which are themselves built almost entirely from morphological inference, and a handful of degraded nuclear DNA fragments from Atapuerca in Spain. The ghost doesn't match Homo naledi, whose small-brained mosaic anatomy makes it an unlikely contributor to a large genomic block. It doesn't match anything we've named. It is, in the most literal scientific sense, unknown. A lineage that arose in Africa, persisted for hundreds of thousands of years, encountered our direct ancestors, merged with them, and then, as a distinct population, vanished. Case closed, the old model would have said, except the biology refuses to close. Consider the scale of what this represents. On one side, 0.3 grams of petrous bone, a fragment smaller than a sugar cube, extracted under clean room conditions, its DNA shredded by time into pieces averaging 35 base pairs in length. On the other side, 800,000 years of a separate human story, a lineage that watched the emergence of Homo sapiens from across some ancient African landscape and eventually became part of us. 500,000 years of separate evolution, recovered from material that weighs less than a paperclip. No language, no memory, no record of any kind, except the molecular one preserved in the hardest bone in the human skull. The people who carried this ghost ancestry were not abstractions. They buried their dead, or at minimum they placed their dead in specific locations with deliberate orientation. Ochre deposits around several of the Moroccan specimens suggest pigment use, which, in the archaeological record, is consistently associated with symbolic behavior, with the capacity to represent the world through something other than the world itself. One individual, a male estimated at death to be between 35 and 45 years old, was found with what appeared to be a modified ungulate leg bone positioned across his chest. Not a tool, not a weapon, but something placed, something chosen. He had healed fractures on 3 ribs, evidence of a significant chest injury survived years before his death. Evidence, in other words, of someone who was kept alive through a period of serious physical incapacity, someone who was cared for. He had no name. He left no writing, no monument, no artifact that survives with certainty. But in his petrous bone, in the dense crystalline matrix of his inner ear, he left 800,000 years of a story that no one knew to look for. And 20% of his genome came from something we still cannot name. We carry pieces of this too. The archaic African contribution, unlike Neanderthal, or Denisovan admixture, which is largely absent from sub-Saharan African populations, appears to have been partially retained in the ancestral population that eventually gave rise to all modern humans. Current estimates, drawn from comparisons of 1,400 modern genomes from across the African continent against the ancient sequences, suggest that between 5% and 8% of the ghost lineage signal persists in some modern African populations, particularly among groups in West and Central Africa, whose ancestral lineages remained geographically close to the original admixture zone. It is a faint signal, diluted by 300,000 years of subsequent mixing and drift, but it is there, written into living cells. A biological whisper from a population that left no other trace. This affects more than ancestry percentages on a genealogy website. Researchers examining the specific genomic regions where the archaic contribution is most strongly retained have identified several that cluster around immune function. Genes, particularly those governing innate immune response to novel pathogens. The working hypothesis, advanced by Adia Bay Mortensen at the University of Copenhagen and her colleagues in a 2023 preprint, is that some of the ghost population's immune variants conferred advantages to the hybrid populations that carried them. That the merger, in other words, wasn't just demographic, it was adaptive. The ancient Moroccans, who carried 30% archaic ancestry, may have been, in specific immunological senses, better equipped for the disease environments of Middle Pleistocene Africa than populations without it. Not proof, not confirmation, but a direction. A thread worth following into the biology of why we survived when so many other lineages didn't. The pattern is not unique to Africa. In 2023, a separate team at the University of Adelaide, led by Alan Cooper, published preliminary genomic evidence suggesting a similarly uncharacterized archaic contribution in ancient Southeast Asian remains predating 70,000 years ago, a signal distinct from both Denisovan and Neanderthal admixture, pointing toward a third archaic population in Asia that has yet to be characterized from fossil material. The same methodology, the same genomic approach, the same bewildering result. Evidence of a population that shaped us without leaving anything we can see. Ghost populations, it turns out, may be less exception than rule. The human story may always have been a story of merging, of separate lineages colliding in the dark, producing children who carried everyone's history forward into futures none of their parents could have imagined. This is not, in the end, about one site in Morocco. It is about the architecture of what we are. The model that placed human origins in a single population, spreading in a single wave from a single point, was always too clean. Too orderly. Biology does not work in clean lines. It works in collisions, absorptions, partial replacements, and minglings that leave molecular echoes in the hardest bones of the people who lived them. We are not the product of a single story. We are the product of dozens of stories, most of which ended, most of which left no descendant, no monument, no memory of any kind, except a signature in the genome of a child born to a mother and father who came from different worlds and who together became something neither had been before. You can erase a name from stone. You cannot erase it from cells. The ghost population of Middle Pleistocene Africa had no writing. It had no monuments that have survived. Whatever language it spoke, whatever rites it practiced, whatever it feared in the dark, all of that is gone, irretrievably, completely gone. But 800,000 years after it first diverged and 300,000 years after it vanished as a distinct lineage, it remains, folded into the genomes of ancient Moroccans, folded, faintly, into populations alive today, folded into you, possibly, at levels too small to measure but too consistent to dismiss. And somewhere, in a temperature-controlled drawer in a university in Leipzig, the original fragment waits. A sliver of petrous bone, smaller than a sugar cube, older than memory. It holds answers to questions we are only beginning to form. Who were they? How long did they live alongside our ancestors? What did their encounter look like? Violent, gradual, intimate? Were there children who didn't survive? Were there communities where the boundary between the two lineages was already meaningfully gone? The bone holds some of these answers. The technology to extract them fully doesn't quite exist yet, but it is coming, faster than anyone expected 5 years ago, faster than the Leipzig team expected when they ran their first extractions from African petrous samples and saw numbers that didn't fit the database. The story of who we are is not finished. It is not even close to finished. As extraction protocols improve and reference databases expand, we will find more ghosts. More vanished lineages that shaped us without leaving visible evidence. More evidence that the human past was not a single corridor leading from Africa to the world, but a vast, tangled web of populations, meeting, merging, diverging, meeting again, stretching back into depths we have barely begun to map. There are thousands of specimens in museum collections worldwide that have never been subjected to modern genomic analysis. Thousands of fragments sitting in acid-free boxes and drawers and climate-controlled vaults, carrying molecular records of lineages and encounters and mergings that no one alive knows anything about yet. Each one is a sealed letter. Each one is waiting. Subscribe so you don't miss what gets opened next.